would you mind coming up and having a seat with Andrea? Thanks a lot, Andrea, for raising a number of really important questions about why we're all here today and what the meaning of epigenetics is in the larger constellation of not only mechanisms, but also why we care about them. So we do have just a few minutes for questions from the audience. So again, please stand up, state your name, where you're from, why you're asking the question, and the question, briefly. Kevin.
Kevin Grove, Oregon Health Sciences University, part-time academic, part-time other. The question, I mean, epigenetics is a really hard one to get your hands around as far as long-term impact, even though I understand that that's what it's really supposed to mean. My question is really around different tissues and different cells. So how common is epigenetic phenomena or methylation or acetylations to be tissue specific? And also within a single tissue, let's say for a liver, where you think you have most of the cells in that liver are similar or homogeneous, how homogeneous is the epigenetic modifications of those cells? How heterogeneous? Is that part of the diversity in the responses?
Yeah, certainly there are the differences. You know, I can speak most knowledgeably about DNA methylation, but as that I showed in that slide by you know, Kundaji et al., that you know that you can look at almost any type of epigenetic mark and find dramatic differences between different tissues and certainly between different cell types. Regarding the liver, I don't know how much has been done at looking at, you know, heterogeneity of epigenetic regulation across, you know, the different hepatocyte populations, but I would guess that there would be differences in the, you know, the periportal versus the perivenous regions and that sort of thing. But I can say that I can bring up one of a recent study of ours from just last year, which is also relevant, in which we looked at the hypothalamus in mice and looked at DNA methylation changes that occur from birth to weaning at 21 days of age, and we separated and looked independently at hypothalamic neurons and glia, and found, you know, dramatic differences in the epigenetic changes occurring during that early postnatal period. In particular, we found much more increases in methylation in neurons rather than in glia during that period. So, I mean, even within a specific region of the brain, there are many different cell types, and of course, even within the neuronal population, I'm sure that you know, the next step would be to look at the different classes of neurons in the hypothalamus, and I'm sure we would see dramatic differences there as well.
As a quick follow-up, though, is reversibility, is that due to cell turnover, like in a tissue like the liver, where you get a lot of turnover in cells and skin, versus the brain where you don't? Is that reversibility tied to cell turnover?
Yeah, absolutely. I mean, there's two potential ways to reverse. Again, focusing on DNA methylation, you can have there is known active demethylation now via a TET-mediated process, but also as you mentioned, just the failure to maintain established methylation patterns during mitosis is another way to have erasure.
I wanted to add to, I think there was another question about whether within tissue there are differences. So methylation is an all or nothing process in a cell. But in a tissue, in fact, the way we measure methylation is proportion of cells that are methylated. So we see a lot of variation, actually. And this is very important because the way we interpret data, we express usually methylation as a % of cells that are methylated. You see a lot of difference between subjects. Exactly.
Hi, my name is Charlotte Pratt from the Blood Institute. I think this is working. My question is for Robert Waterland. Can you kindly elaborate on the whole issue of dietary methyl donors, and specifically which ones have you used in your study?
Okay, yeah, I didn't spend a lot of time on that, But in our studies, our studies with the AVY mice and subsequent studies in related models, we've used a methyl donor cocktail, if you will. We supplemented the mouse diet with extra folic acid, vitamin B12, betaine, and choline. And I often get asked this, well, will folic acid alone do the same thing? And we have some unpublished data showing that no, it will not. So some combination of these nutrients is required to cause this pro-methylation effect in the offspring. And getting at it from another angle, we recently published a paper in collaboration with Andrew Prentice, where we were able to show how a mother's nutritional status around the time of conception affected DNA methylation at metastable epialleles in her offspring. And so you might check that paper as well, which essentially corroborates that one carbon metabolism is playing a key role.
We have time for 2 more questions. Caroline and Judith.
Judith Hall, University of British Columbia. You mentioned cloning, and I wanted to extend that a little bit and ask about assisted reproductive technologies and monozygotic twins, particularly monozygotic twins, because they've been used to show a genetic relevance for obesity. And it seems to me that both of those run some risk of not having normal epigenetics.
Yeah, the issue of assisted reproductive technologies is clearly very important, and I find that just amazing that there seems to be more regulation about what goes on into our breakfast cereal in the morning than what is the specific composition of the media that are used for these early embryos during the in vitro fertilization process. So, yeah, I think there are some early studies coming out suggesting that that assisted reproductive technologies, that individuals that are conceived in this fashion run a higher risk of certain developmental diseases, but it still remains unclear, I think, whether or not those epigenetic diseases associated with assisted reproductive technologies, whether they result from the process itself or could actually be a result of epigenetic aberrations that were present in the sperm or the egg and contributed to infertility. So those are some questions that are still outstanding. And in terms of MZ twins, yeah, that's a very interesting point that you raised. That we are looking at some of these regions that we've identified as candidate human metastable epialleles, and what we sometimes see is that even though genetic data indicate that genetics is not influencing the establishment of methylation at these loci, we see very high concordance within MZ twins. And our interpretation of that is that even if the epigenetic state is established before embryo cleavage, then you could have the exact same epigenetic state in the 2 twins, not because of their genetic identity, but simply because of developmental timing. And I think those kind of stochastic effects, if they're occurring in the very early embryo, MZ twins will be the completely wrong model to study those in.
One more.
Caroline Relton from the University of Bristol and Newcastle in the UK. My question relates to the issue of persistence of DNA methylation changes that are incurred during early development. And you, Rob, very eloquently alluded to this notion of, you covered Alan Lucas's early work talking about metabolic and biological programming, which stated that an early insult sets in train a sequence of events which culminates in disease in later life. So my question really is around whether or not epigenetic changes fall into that broader paradigm. We look sometimes quite considerably later in life, in adulthood, for epigenetic marks that infer early life exposures. And perhaps those epigenetic marks have already disappeared by those points in later life. And whether, in fact, in our own work, we see changes that are evident in cord blood at birth attenuate very rapidly in early childhood. But that doesn't mean they're unimportant in relation to development of subsequent disease. They may set in motion a physiological chain of events which influence obesity or other diseases. And I wondered how you feel that fits with how you presented this notion of persistence and looking for persistent epigenetic marks across the life course.
So I think your question is whether it's really clear in humans whether specific DNA methylation marks are established during early development and then persist into later life. And I mean, clearly that is the case in terms of with these metastable epialleles in the mouse studies. We could show that that we could measure methylation early in life at these loci and that and in adulthood and find the exact same level and exact same degree of inter-individual variation. And in again, in our recent study that was published last year in Nature Communications with Andrew Prentice, we showed the same thing in humans as well. That we can also no, I'm misspeaking. These are data that are a paper that is under review, where we do have data now in humans where we show longitudinal data at least one of our regions that we've looked at, showing that methylation at age 7 predicts that at average age of 17. And so we are able to show persistence at these metastable epialleles. But your point is well taken, that just measuring some epigenetic differences in adulthood and relating that to an earlier exposure is not clear whether those epigenetic changes are causal or some kind of secondary effect.
Thank you very much, Rob and Andrea, for your wonderful talks. I'll leave us with the second part of Caroline's question for the rest of today and tomorrow, which is even if DNA methylation or other epigenetic processes are transient, could they set in motion a programming phenomenon? Thank you very much, both of you.